I and my colleagues were studying the interactions between gut microbes and the diet to look at their impact on childhood undernutrition and growth of the mammalian host. And we do this primarily in mouse models, and I'll tell you about a new model that I helped develop in notobiotic piglets. So the key focus of our work is on childhood undernutrition. And to give you a little bit of a background, undernutrition or malnutrition is a global health crisis. It accounts for over 3 million deaths annually, which represents about 45% of death for children around the world under the age of 5. And in cases where malnutrition is not fatal, it's associated with long-term side effects that include stunted growth, impaired cognitive development as determined by intelligence testing, and immune dysfunction. To show you a bit of data on this, this is the incidence of stunting in the continent Africa from 1990 projected out through 2025. And what I hope you can see is that although the prevalence of stunting has decreased slightly and is projected to continue going down, the absolute number of children with stunting is increasing over time, and we feel this is unacceptably high. Importantly, we should look at existing nutritional interventions that have been used for treating malnutrition, and what we find is that the effect of these interventions has been modest and in most cases temporary. So new avenues of treatment are needed. When we think about the factors that underlie nutrition, and in particular malnutrition, we think about certain salient features that include the diet that the child is eating, their immune system, and also the presence of known enteric pathogens that can inhibit growth and cause disease. But something that lies at the center of all of these factors is the gut microbiota. And so when I say gut microbiota, I'm referring to the community of microbes that inhabits the human intestine that assembles beginning at birth, so children are born germ-free essentially, and over the first few years of life, that community assembles into an adult-like configuration. This community is dominated by bacteria, and it's known to play critical roles in the degradation of food ingredients, the maturation of innate and adaptive immunity, and providing a barrier to enteropathogen invasion. So some of the key questions that we were interested in addressing were, first, does the microbial community in the gut follow a developmental program in the first years of life that's generic across different hosts and different populations? And in cases where you see impaired nutrition and growth, is that undernutrition associated with disrupted gut microbiota assembly and function? And lastly, is the gut microbiota, if this is the case, which turns out it is, does the gut microbiota play a causal role in host growth outcomes as opposed to strictly being correlated with them? So to answer the first question about how does the gut microbial community assemble, I'll tell you about some work from my colleague Satish Subramanian, another graduate student in Jeff Gordon's lab. So Satish teamed up with our collaborators in Bangladesh to collect fecal samples from children from the ages of 0 months up to 24 months of age, and characterize those samples for their gut microbiota composition using phylogenetic marker gene sequencing, and the gene that they were sequencing is the 16S ribosomal RNA gene. And in doing so, he could identify which species were present and in which proportions in these samples over time. So the data here I'm showing you is sort of an average across that whole population of healthy infants. On the x-axis here is time, starting with the youngest samples up to 2 years of age. And each row in this heat map represents a bacterial strain that Satish detected, with the color indicating the abundance of that strain. And so what I hope you can appreciate is that over time, there are differences in terms of the abundance of particular strains as the child ages. So some strains are more abundant early in life and sort of fade out, whereas others are more abundant later in life and are not detected shortly after birth. In other words, this community follows an ordered program of assembly across healthy infants. Satish took this one step further, and he used an ensemble machine learning-based approach called random forests to identify a set of 24 age-discriminatory bacterial taxa, and those are the 24 that are shown here, in healthy twins and singletons from Bangladesh. Using these 24 strains, Satish developed a model where he could predict, based on the configuration of the microbiota sample, the age of the child, and this he termed a microbiota age, right? So that's predicted based on which bacterial species are present and in what abundances. And he found that this microbiota age was highly correlated to the chronological age of the child, and that's what I'm showing you here for healthy children. But he wanted to look at how microbiota age, or this maturation of the microbiota, was impacted by malnutrition. So he profiled the microbiota from a set of Bangladeshi children from the same population over time that were malnourished or stunted. And what he found is that the predicted microbiota age using this same model developed using healthy children showed that the microbiota age, or the predicted microbiota age, was lower for children that were undernourished as compared to their healthy counterparts. In other words, these children harbored a persistent gut microbiota immaturity relative to their counterparts. It's also important to note that Satish took this one step further and examined gut microbiota development and maturity in a second population of Malawian children, and found this same pattern where undernourished children had an immature microbiota relative to healthy children, indicating that these observations are not limited to a single population, but are in fact generalizable to other dietary and cultural contexts. So with those observations in mind, we wanted to ask the question of whether the gut microbiota is not only correlated to host growth, but whether it plays a causal role in host growth and development. So in order to do this, we turned to notobiotics, and I'm very grateful for the earlier introduction of notobiotics. That was very thorough. In our case, we're using notobiotic mice. So these are mice that are born and maintained germ-free and kept inside the sterile environment of these plastic vinyl isolators that I've shown you on the right. And these mice can be maintained as sterile throughout their entire life if you want to, or in our case, they can be selectively colonized with the communities of interest to examine their interactions with the diet and their effects on host growth. Importantly, notobiotics allows the replication of a single human microbiota across multiple recipient animals for experimental purposes. And again, this allows us to examine the interactions between food, the gut microbiota, and its effect on the host animal. So together with my colleague Laura Blanton, another graduate student in the lab, we teamed up to answer the question of whether the nutritional status of the host had an effect, and the corresponding microbiota configuration had an effect on host growth outcomes. So in order to do this, we took groups of 5-week-old germ-free mice. So these are young mice that are still actively growing, each group housed in their own notobiotic isolator to prevent the exchange of microbes between them. And each group was colonized with a single microbial community from either a healthy or a stunted Malawian infant. The mice were fed a sterile diet representing foods commonly eaten by the Malawian microbiota donors from which they were sampled. And then we monitored the growth of these animals over a course of about 5 weeks from the time they were colonized, along with their body composition and the gut microbiota structure in these animals. So the growth trajectories of the two groups are plotted here. On the x-axis is time, days after an introduction of the microbial community, and on the y-axis is the increase in weight normalized to the start when we introduced microbes for the first time. And what you can see is that mice that harbored a microbiota from a stunted or an underweight donor gained significantly less weight over time than those that harbored a healthy infant's gut microbiota. Importantly, when we look at the body composition of these animals over time, we find that this difference is accounted for in the lean body mass of the animals. So that's everything except for fat, muscle, bone, connective tissue. In fact, we found no difference at all in fat mass gain between these two groups of animals. And we found no difference whatsoever in their food consumption. So although these mice have the same genetic background, they're fed the same diets, and they're eating approximately the same amount of food, those that harbored an undernourished microbiota gained less weight and less lean body mass over the same period of time. So Laura took this one step further and took a similar approach to what Satish did, and used a random forest-based model to identify a set of growth discriminatory bacterial taxa, many of which overlapped with the age discriminatory taxa that Satish had discovered. She then designed an experiment to directly test whether these growth discriminatory bacteria could improve growth in the context of animals harboring a stunted or underweight prototypical microbiota. So this is one from the original screen that led to low growth, and she's now adding in growth discriminatory taxa cultured strains in a consortium of 5 of them to see whether these strains could improve or rescue the growth phenotype that was absent in the undernourished community. And what she found is that administration of this 5-member consortium of growth discriminatory strains significantly improved both body weight gain as well as lean body mass gain in the recipient animals. And again, it had no impact on the fat mass gain of these animals. As a technical note, only 2 of the 5 strains, Ruminococcus navis and Clostridium symbiosum, successfully were detected in the fecal microbiota of these mice after administration. But this still serves as an important proof of concept that administration of these growth discriminatory strains led to increased growth in the recipient animals. All right, so just to recap this first half of the talk, my colleagues have found that assembly of the infant gut microbiota in Bangladeshi and Malawian infants follows a pattern of ordered assembly, and that this pattern of assembly is disrupted in children that are undernourished. We then went further and found that transplantation of undernourished versus healthy Malawian microbiota into germ-free mice transmitted discordant growth phenotypes to the recipient animals. And furthermore, that the growth of mice harboring an undernourished microbiota could be augmented by administration of growth discriminatory bacterial strains. So this transitions into what I was interested in looking at with my own work, which is understanding how infant gut microbes interact with first foods that an infant encounters and how this interaction functions to promote host growth. So when we think about what important first foods there are for infants, the first to come to mind is probably breast milk. So the World Health Organization considers this breast milk the optimal nutrition source for growing infants and recommends exclusive breastfeeding for the first 6 months of life. Exclusive breastfeeding is associated with several known health benefits, including reduced infant mortality, fewer infections, and improved neurodevelopmental outcomes as determined by intelligence testing. But what are the underlying mechanisms of those benefits, and how specifically do microbes, gut microbes, impact the nutritional value of breast milk? When we break down what's present in breast milk, and here I'm showing you a breakdown of the solid components, we find that it's dominated by lactose, lipids, and proteins, along with a 4th component called a class of compounds called the milk oligosaccharides. And it's important to note that existing nutritional interventions targeting malnourished children have focused on lactose, lipids, and proteins. So my key area of interest was on this class of compounds called the milk oligosaccharides, which are structurally diverse carbohydrates. Over 200 unique structures have been identified so far by mass spec methods. And importantly, milk oligosaccharides are commonly thought to be indigestible by the host animal. They can, however, be readily utilized by many members of the infant gut microbiota. So it stands to reason that effects of milk oligosaccharides on host biology may involve or be mediated by members of the infant gut microbiota. As an important note, many human milk oligosaccharides are decorated with residues of fucose and or silac acids. And silac acids are actually a class of compounds, but the most abundant of them is this N-acetylnuraminic acid, which I'll just refer to simply as silac acid throughout the talk. But silac acid is thought to be functionally critical both in the gut as it is incorporated into host mucus glycans, as well as in the central nervous system of a growing animal. What's interesting is that the activity of the rate-limiting enzyme for synthesis of this molecule is actually limited in the neonatal period. So it's thought that external sources of silac acid are important for its incorporation into host tissues. So the key questions I was interested in addressing with my studies were, does the gut microbiota make HMO conjugated silac acid available to the developing infant? And more broadly, how does the HMO content of breast milk, including sialylated HMOs, relate to infant growth outcomes? To address the latter, we teamed up with our collaborators in Finland and Malawi and the University of California, Davis to characterize breast milk from Malawian mothers in 2 independent cohorts. The first cohort had 88 mothers and the second had 215. The milk samples were collected from these mothers at 6 months postpartum and characterized for its HMO content by mass spec methods. And the mothers were then binned by the growth outcomes of their children, either healthy or severely stunted. And what I'm showing you here is the abundance of milk oligosaccharides in those breast milk samples when the mothers are binned by the health status of their infant, either healthy or stunted, and split by the 2 cohorts. And what I hope you can see is that both total, fucosylated, and sialylated milk oligosaccharides were more abundant in mothers of healthy children than in mothers of the stunted children. And this was true across both independent studies in Malawi. It's important to note, I think, that this difference is relatively modest. These are normalized to the amounts present in the healthy children. But this small difference could represent a large cumulative difference in dose that the children are observing over the long period of exclusive breastfeeding. So from my own work, I wanted to experimentally address whether sialylated milk oligosaccharides promote host growth, and do the effects of milk oligosaccharides depend on gut microbiota. So again, I turned to Notobiotics with a couple of small extensions to the model I described to you before. Firstly, I needed a Malawian infant microbiota to serve as the backdrop for these milk oligosaccharide supplementation studies. And in order to do that, I used a 25-member consortium of cultured strains isolated from a severely stunted Malawian infant and representing the majority of diversity present in that community. So this is an unhealthy background. The mice were again fed an 8-component Malawian diet, which we call M8, that is representative of foods commonly eaten by Malawian children in this population. And thirdly, I needed a source of milk oligosaccharides for supplementation to test their effects and their interactions with microbes, which turns out to be a little more challenging than you might think. So in order to get a source of milk oligosaccharides for these experiments, as you might imagine, large-scale isolation of human or mouse milk oligosaccharides is not really a feasible endeavor. But bovine milk, or cow's milk, contains milk oligosaccharides that can serve as a source for these experiments. Importantly, bovine milk oligosaccharides are structurally similar to HMOs, and they can be derived from existing milk production waste streams, which means that they would be scalable for follow-on studies in the clinic if it went that way. So thanks to our collaborators at UC Davis, David Mills and Daniela Brile, we were able to get our hands on this mixture of sialic acid enriched bovine milk oligosaccharides, or SBMO. The composition of the milk oligosaccharide composition of that mixture is here. And what I just want you to take away from this is that most of this mixture is composed of this one compound, sialolactose, which is this triose containing sialic acid. This SBMO was added directly into the Malawi 8 diet, and importantly, by nutritional analysis, the comparison comparing the control diet to the supplemented diet, they were isocaloric. In other words, differences that we see in growth of these animals can't be attributed to gross differences in caloric content. So the experimental design follows something similar to what I told you about with my work with Laura, wherein groups of 5-week-old actively growing germ-free mice were colonized with a 25-member community from an unhealthy Malawian infant. I determined the colonization efficiency by shotgun sequencing and found that about 75% of the strains effectively established in the mouse gut. And these mice were then fed the Malawi 8 diet with or without the addition of SBMO over a period of 5 weeks. As a separate control, I included a group of mice that were fed Malawi 8 diet supplemented with inulin. Now, inulin is a fructan, or a polymer of fructose, that is commonly added into infant formulas with the express purpose of mimicking milk oligosaccharides, but it is structurally distinct from milk oligosaccharides, and I think it serves as an important comparator in this study. So here's the upshot of what happened with these mice. Again, on the x-axis is the time since the initial colonization, and on the y-axis is the weight normalized to the start of the experiment. And what you can see is that mice supplemented with the SBMO, or the sialylated oligosaccharides, gained significantly more weight than the mice fed the control diet alone. Importantly, mice supplemented with inulin, so this is the structurally distinct fructan, do not have this same augmentation of growth. Importantly, I observed no difference in food consumption between these groups of animals. When I performed a similar experiment, which I think is an important control here, in mice that were never colonized, these mice follow the same timeline, but they're germ-free throughout, I saw no difference in the weight gain between mice fed the Malawi 8 diet alone and the Malawi 8 diet supplemented with SBMO, which importantly tells me that this SBMO-mediated growth phenotype is dependent on the presence of the gut microbiota. Also, when I look at the body composition in this experiment of the animals, what I find is that the difference between the control animals and those supplemented with SBMO is accounted for in the lean body mass of the animals, similar to what I showed you in the studies with Laura. Importantly, there is no difference noted in the fat mass of these animals, so they're not simply getting fatter as a result of this treatment. So I mentioned that sialolactose represents the majority of the milk oligosaccharides in this mixture. And I also told you that sialic acid is thought to be very important for brain development. And as a point in that direction, I guess a 10-year-old study now showed that supplementation of newborn piglets with sialylated glycans improved both their memory and their learning abilities. So this is getting right to the point, but what I found is that the free sialic acid in the brains of these mice, that was measured by GCMS, was significantly higher in mice that were supplemented with SBMO than in control animals, which suggests that not only are these animals growing more as a function of treatment with the sialylated milk oligosaccharides, but that the sialic acid present in the mixture is bioavailable to the host for incorporation into their tissues. All right, so for just this last bit, I'd like to tell you about a bit of a translational model that I worked on towards the end of my PhD, trying to answer the question of whether these observations that we saw in mice translated to a second species, namely pigs. So we developed a model of using notobiotic piglets, primarily because pigs are more closely related evolutionarily to humans and they share more anatomical similarities to humans than do rodents. And these pigs also grow they're domestic pigs, they grow very rapidly in their first weeks of life, which we feel gives us a better chance of seeing phenotypic differences as a result of treatments. These pigs were delivered by cesarean section directly from the pregnant sow into these notobiotic isolators. Here's an example of one here. So you've got the traditional vinyl isolator on top of this sterilized steel cage. And here's one of the pigs at about 3 weeks old. So you can see they get pretty unmanageable pretty quickly. The experimental design is fairly similar to what I did in mice. So again, the pigs were delivered by cesarean section, and their sterility was confirmed over the first few days of life, but within isolator actually, by culture. The pigs were colonized at 3 days of age after the sterility was confirmed with a 17-member defined Malawian infant community. This is the same community I used in mice, but I just removed some of the pathogenic components because it's known that gut barrier function in newborn pigs is very poor, and we didn't want to take a risk with the animal safety. So this reduced community was used, but it still represents the majority of diversity present in the donor unhealthy Malawian community. The pigs were fed a sow's milk replacement formula initially, but then were transitioned onto the same Malawian diet that the mice were fed for those experiments. And at the time that we completed weaning of these animals, which was at the ripe old age of 11 days, half of the animals were switched from the Malawi 8 control diet to the Malawi 8 diet supplemented with SBMO for the remainder of the experiment. And again, what we found is that supplementation of the pigs with SBMO promoted increased weight gain over time compared to those fed the Malawi 8 diet by itself, indicating that the observation that SBMO, or these sialylated milk oligosaccharides, promote growth was true not only in mice but also in pigs. Importantly, this is a single experiment. This result was reproducible across replicate experiments in the notobiotic piglets, showing that not only did it translate, was it reproducible within a model, but across models. So in conclusion, what I hope to have convinced you of is that Malawian mothers with undernourished infants have decreased milk oligosaccharides in their milk, and that in animal models, both mouse and piglet, SBMO, or this mixture of sialylated oligosaccharides, promoted growth in a microbiota-dependent way, in a structure-specific way. This effect was not seen with fructan inulin, and it correlated to increased free sialic acid levels in brain. I don't have time to go into more detail, but we found that there were lots of other effects of SBMO on host biology, including differences in bone morphology, differences in metabolic response to the feeding and fasting transition in our mice, and marked differences in bacterial gene expression that helped to explain some of what's happening mechanistically. And again, this was translatable to a second model animal. So I have just a few people to acknowledge. First, my PhD advisor, Jeff, at WashU. The larger consortium, the Breast Milk Malnutrition and Immunity Consortium, our whole network of collaborators, which I hope to have represented here, and our funding agency, the Bill and Melinda Gates Foundation, which funded the whole BMMI project and all of the work that I've shown you here today. So with that, I'd be happy to take any questions.